Hello and welcome back to our discussion of epithelial tissues. As for the last video, I talked about the types of epithelia we have, and even before then, I talked about the fact that epithelial tissues are lining tissues. But we said that some of these lining tissues are just for covering, and then some others are for secretion. By the way, we didn't mention in that video that the functions of epithelial tissues are numerous and they include the following. I've put the functions here in an acronym that you try to remember. S, scared, where S means sensation, the other S means secretion. C means cleaning. A is for absorption. R is reduction of friction. E is excretion, and then the D stands for diffusion. So those are functions of epithelial tissues. Just in case you find that in the exam, remember to eliminate the one that is not on this list. Now beyond that, we already said that secretory epithelia are usually found in structures called glands. So we'll need to talk about glands very briefly. I'd like to talk about glands. And generally, we know that glands are structures that secrete substances. And we have different types of them. We have some glands that we refer to as exocrine. And then we have the ones we call endocrine. And then there's a third group that we refer to as paracrine. Now, what's the difference between the exocrine, the endocrine, and the paracrine glands? I can establish the difference just by showing you how the gland communicates with its target. What do you mean by target? The organ and a secretion acts on is a target organ. For example, if I were a gland, and I were to make a chemical that would work on you, then you would be called my target or the target of my secretions. So it means that for a gland, usually, when materials are produced, they have to be transported to a certain point where they carry out their function. So everything now depends on how the secretions finally reach the target. In one case, when a gland finishes the process of secretion, it will send out the secretions to the target through a tube like that. So there's a tube connecting the gland to the target. If you don't call this a tube, you can call it a duct. But there's another case where you have a gland, G, and then you have the target here, T. Between them, there's no tube. Instead, when blood is transported around the body, it is possible that a blood vessel passes beside the gland like this, and that same blood vessel, or a different blood vessel as a matter of fact, can pass through the target, so that the secretions within this gland are sent into this blood vessel, and since blood spreads around the entire body, that blood can reach this target organ here and then move into the target. So at the end of the day, the secretions have gone from gland to target, not by any tube, but through the blood. The last case is one where you have a gland very closely situated to its target. When gland and target are very close, like you can see here, it's possible for the secretions of the gland to move straight into the target by simple diffusion. This case, where gland and target are very close and can communicate as you have seen there, is where we refer to the gland as paracrine. And paracrine glands are commonly found, not so commonly though, but you can have them in the gonads, like the testes, the ovaries. Testosterone works in the testes, and where is it produced? Right there in the testes. So that's an example of paracrine glands, the gonads. Then the case where the gonad communicates with its target through the blood, no tube joining them. we say that the gland is endocrine. If you don't want to use the word endocrine, you may also call it a ductless gland. Why is it called ductless? Because it doesn't have any duct or tube. That case where there is a tube connecting the two of them is where we refer to the gland as an exocrine gland. So that's the essential difference between exocrine, endocrine, and paracrine. Next, I'll be talking about the types of exocrine glands we have. So the next part of this discussion will focus mainly on exocrine glands, how we have different types of them, and what differentiates these exocrine glands that we'll be talking about. So to differentiate exocrine glands, I've put a small outline here to guide me and to guide you. Exocrine glands can be differentiated on the basis of one. I didn't mention examples of exocrine and endocrine glands, just in case. The glands we have in our digestive system that make enzymes are exocrine. Then you can think of the sweat gland, think of the mammary gland, think of the meibomian gland, many other glands like that in the body are exocrine. Then the glands that you know that produce hormones, like the pituitary, the thyroid, parathyroid, the adrenals, all of those are endocrine glands. They don't have ducts. But it's important to mention that the pancreas has an exocrine portion as well as an endocrine portion. So one gland that is both exocrine and endocrine is the pancreas. Away from that, types of glands. The first basis on which we are going to classify glands is the number of secretory cells. Number of cells. There are some glands that are made up of just one cell, a single cell acting as a gland. Then you also have some glands that are made up of more than one cell. We say those ones are multicellular glands. Now for the unicellular glands, we don't have so many examples, but the goblet cells that are found in the intestines, we find the goblet cells in the intestines, in our digestive tract those goblet cells are able to function as glands even though they are single cells. So we say that they are unicellular glands. Then the other glands that we have commonly are multicellular. So like I already said, unicellular glands are not common, but multicellular glands are everywhere. Even though unicellulars are not common, do we have an example? Yes, the goblet cells. So away from that, classification according to number of cells. The next thing here is classification according to the nature of the secretion. Some glands, okay, so on the basis of nature of secretions now, there are some glands we refer to as serous glands. Then there are some glands we refer to as mucous glands. Then we have some glands that we refer to as mixed. And then finally, we have the ones we refer to as miscellaneous. When we say a gland is serous, it means that the secretion of this gland is like serum. If I would make that way easier, I would say that the secretion is thin, like water. Thin, very thin secretion, not thick. So the secretion is like water, that's a serous gland. Then a mucous gland has secretions that are thick. All right, they have a thicker consistency. You can think of mucus, normal, that we know, that we bring out phlegm. You see how thick that is. So mucous secretion, due to the presence of what we refer to as mucin. So there's mucin in mucous secretion, which makes it have that thick consistency. Then for the mixed glands have portions. A mixed gland is a gland that has a region that has mucous secretion and another region of the same gland that produces serous material. Then finally, miscellaneous. The miscellaneous glands, their secretions are neither serous nor mucous, but they have their own nature. Let's say some more things about them. For the serous glands, their cells are usually polyhedral, so they have polyhedral cells, and then they have a nucleus that is centrally located nucleus. But down here, the mucous glands, the nucleus is usually not central. Instead, the nucleus is basally located. So what do you mean by basally? That is at the bottom of the cell. So when you look at their cells, you can actually differentiate serous from mucous. Apart from the position of the nucleus, they both show polarity. That is in terms of how their organelles are distributed. Their organelles are not just scattered around the cells. Instead, you have certain organelles like the Golgi bodies at the top of the cell. Then at the bottom, you could have the endoplasmic reticulums and so on. So it's just like you're looking at a cell this way. Certain organelles are here, certain other organelles are there. Nucleus in the center. This is the typical arrangement for serous cells. Mucous cells have a similar arrangement, but the nucleus is not central. We said the nucleus is at the base. All right, so all of those details may not be very necessary, but what you must remember is, like we said, serous secretion is like water, is watery. The mucous secretion is thicker. For serous glands, do we have any example? Yes. The parotid glands. If you remember, the salivary glands are of three main groups. We have the parotid, we have the submandibular, and then we have the sublingual glands. So submandibular glands, sublingual glands, and parotid glands. Those are the three groups of salivary glands. Of these three groups, the parotid glands have secretions that are entirely serous, 100% serous. So we say parotid glands are good examples of serous glands. They have serous secretions. Apart from the parotid, the pancreas is also an example there. So pancreatic secretion is usually serous. Then for mucous glands, an example is the Weber glands that we have on the tongue, Weber's glands. So the Weber glands on the tongue are a very good example of mucous glands. Then for mixed glands, that is a gland where a portion produces serous secretion and the other portion produces mucous secretion, we have the other two groups of salivary glands as examples there. So the submandibular gland is mixed and the sublingual gland is also mixed. But when we say they are mixed, like the submandibular, it is something like this. Let me just use this structure to explain it. So this is the submandibular now. So there is a large portion and a small portion in terms of quantity of secretions. The submandibular gland is largely serous, which means it produces a large amount of serous secretion and a small amount of mucous secretion. That is the submandibular for you. But for the sublingual gland is predominantly mucous. So the larger portion of this secretion is mucous. Then there's the small portion that is serous. And for such glands like the sublingual glands, we may want to represent it this way. Look at this now. Let this represent the sublingual gland. Something like that. Such that this region is for mucous secretion. So whatever comes from here is mucus, and then this top is where serous secretion comes from. So you see that the serous portion is very small. It's like a cap, a small cap on top of the gland. So we refer to them as the serous demilions. So the serous demilions are like small caps. They are like serous caps sitting on top of the main gland, which is mucus. So if we must put submandibular and sublingual into categories 1 and 2, then submandibular will be put under serous, whereas sublingual will be put under mucus. But in reality, the two of them are mixed because their secretions are partly serous and partly mucus. Then for miscellaneous glands, we said those ones have secretions that are neither here nor there, neither serous nor mucus. And if you look at examples of such miscellaneous glands, we have the seminal vesicles. The seminal vesicles in males, what do they produce? They produce seminal fluid. This seminal fluid is neither serous nor mucus. It is described as viscid. We say it's a viscid secretion. So it is miscellaneous. It doesn't fall into any of the categories beyond or before that miscellaneous. Then see this other one. See this one. We talked about the sebaceous glands. You must be familiar with the sebaceous glands in the skin. What do they produce? An oily secretion. Their secretion is oily or greasy. Alright, so those ones are also miscellaneous because oil is neither serous nor mucus. Then we have a good example in the ceruminous glands. Ceruminous glands. These ones are found in the ear and they are the ones that produce ear wax. So we say their own secretion is waxy. So ceruminous glands, sebaceous glands, seminal vesicles are all miscellaneous glands because their secretions are neither serous nor mucus. So I'll leave that classification criterion number 2, which is nature of their secretions. Number 3 criterion would be mechanism. That is how they send out their secretion. Mechanism of secretion. In terms of the mechanism by which they secrete, glands may be classified into one, we have merocrine. Merocrine glands are also called eccrine glands, and they are the majority of glands. Then we also have the apocrine glands, and then beyond apocrine we have the third one, holocrine. So first you need to know the terms used. So in terms of how they secrete, we say glands may be merocrine, eccrine, or apocrine. Now merocrine, apocrine, or holocrine. Now to talk about the meanings of those terms, picture a gland like this. Picture this to be a gland. So what I have here is the duct. Alright, this is the duct system of the gland through which the secretions of the gland will eventually reach the target. So this is the main part of the gland that we refer to as the secretory unit. So we have the secretory unit and we have the ductal system. This is the secretory unit and then this is the ductal system. In fact, beyond this criterion number 4, sometimes we add criterion number 5. Under criterion number 5, we classify glands based on the nature of the ductal system. But this criterion number 4 that I'll talk about is based on the shape of the secretory unit. So based on the shape of this, we have different types of glands. Based on the nature of that, we have different types of glands. That's supposed to be number 5, but I'm leaving that out to end at number 4. So back to number 3, merocrine, apocrine, and holocrine. Let's look within the gland itself now. See what we have in the gland. These things I am drawing are cells. They are the ones that produce whatever secretions this gland is making. So these are cells. Alright, so the secretory unit has been fully drawn. This is the secretory unit. Within that secretory unit, we have many cells, which makes this gland multicellular. If you remember, this is our first classification. We said a unicellular gland is just one cell, but this gland we are looking at now is multicellular because it's made up of many secretory cells. So all these guys are busy secreting right now. When they finish secreting, how will the secretion come out? That's what we mean by mechanism of secretion. In the case of merocrine secretion, merocrine, if you bring one of these cells out, this is the cell, and you remember your normal cell biology, the basics of cell biology. When a cell produces things, when a cell is done secreting things, materials are able to go out of the cell by a process called exocytosis. So during exocytosis, it is as though materials diffuse out of the cell. They pass through the cell membrane, just freely they walk out. Let me express it that way. So the process of materials being secreted out of a cell is exocytosis, which is the opposite of the process of materials coming into a cell, endocytosis. So exocytosis, materials are produced and they are transported out of the cell, maybe through special receptors or transporters on the membrane. So that can happen here, such that materials have been produced in this cell and they get exocytosed. They just go out by exocytosis. In that case, we say that the secretion is merocrine. We say that that particular gland is a merocrine gland because its secretions come out by exocytosis. Once you see that word exocytosis in relation to a gland, the word exocytosis already tells you that that gland is merocrine. So once you hear merocrine, think exocytosis. When you hear exocytosis, think merocrine. For apocrine glands, those ones are glands where the cells, again, so these cells now have secreted materials. This is one of those cells. And the material that has been secreted will go close to the cell membrane. See, the material that has been secreted is to stay close to the cell membrane like this. After staying close to the cell membrane like that, a portion of the cell, will now pinch off like this. Look at that portion. It pinches off and falls off like this. See the portion. So the cell becomes this now. That portion has pinched off. So when there is pinching off as a way of release, that's different from exocytosis. In exocytosis, cytoplasm is not lost. Only the material goes out on its own. But in this case, some cytoplasm is lost. And that case where you have a pinching off is where we refer to the gland as what now? An apocrine gland. So in apocrine glands, there's pinching off of a portion of the cell to release its secretions. Then for the last one, holocrine. Materials do not transport out as in exocytosis. A portion of cytoplasm is not pinched off as you are seeing there. Instead, if this is the cell that has secreted materials, after secreting materials, the cell will fall into the lumen. This is called the lumen, the space in the center. This whole cell falls into the lumen and then breaks down in this lumen. It just breaks down to release its secretion. So in that case, watch. This is the cell. Materials did not leave it. A portion of cytoplasm was not pinched off. Instead, the entire cell fell into the lumen and broke down. Because the entire cell, the whole cell, listen to that, the whole cell fell into the lumen, we'll call the type of gland holocrine. Whole cell, the whole cell falls in. So that makes the gland holocrine. So anytime you see a question that says a type of secretion mechanism in which an entire cell or a whole cell falls into the lumen of the gland and disintegrates, that's holocrine secretion. All right, so straight to the point. In summary now, merocrine secretion is by exocytosis, apocrine secretion is by pinching off, and then holocrine secretion involves the whole cell falling into the lumen and breaking down. Examples of merocrine glands: many of our glands are merocrine. Most glands are merocrine. Like I said, apocrine glands, those ones become, they are glands that have their functions influenced by hormones. Yeah, especially the male hormone, female hormone. So they are glands that become active by puberty. In a small child of maybe 1 or 2, the apocrine glands may not necessarily become functional, but in later stages or later in life, yes, they become very functional. They secrete under the influence of the sex hormones like testosterone, progesterone, so that in terms of their locations to even their secretions, the apocrine secretion is usually fatty. Yeah, it's not like ordinary water. Eccrine sweat from the forehead. The sweat glands we have at the forehead. When you clean sweat from the forehead, it's usually like water. But that's different from sweat that we have in the axilla or sweat that we have in the pubic area. In such cases, the sweat that you find is apocrine sweat. It is not just, its composition differs from what you have in the forehead. That one is eccrine sweat, but this is apocrine sweat. All right. So apocrine, yeah, like I said, the glands secrete in response to the male and female sex hormones. Therefore, by default, the glands become active during puberty. What are examples of apocrine glands? We have several ones. I've talked about the sweat glands in the axilla, that is the armpit, and then the sweat glands that we have in the pubic area. We've mentioned that also. Then you also have the glands around the nipple. Around the nipple, we have some of those apocrine glands. They are present there. Then we also have the ciliary gland. The ciliary gland is also called the Moore gland in the eyes. So you have the ciliary gland or Moore gland. This should be a capital letter. And then you have the ceruminous glands. Ceruminous glands, if you remember them, we said that based on the type of secretion, ceruminous glands are miscellaneous glands. But based on mechanism of secretion now, ceruminous glands are apocrine glands. Then for the holocrine glands, we have examples as well. A good example of the holocrine glands are the sebaceous glands. The sebaceous glands in the skin. And if you remember, we also said that the sebaceous glands are also examples of miscellaneous glands in terms of their secretions, which we said is waxy. Then apart from those ones, we also have the tassel glands. Tassel glands are also present in the eye, near the eyelashes. Tassel glands are also called meibomian glands. Meibomian glands. So the meibomian gland or tassel gland is also an example of holocrine glands. So please take note, around the eyes, tassel glands, ciliary glands. Ciliary, apocrine, meibomian, holocrine. So in terms of mechanism of secretion, those are the types of glands. Let's move on to the last one. In terms of the shape of this secretory unit, not the duct system now, the secretory unit. So in terms of the shape of the secretory unit, of course, we have different types of glands. I'll talk about them here. We can refer to the glands as tubular or acinar or tubuloacinar. So these are the three types of glands on the basis of the shape, not of the ductal system, but of the secretory unit. Now, when we say a gland, let me add this, acina is also called alveolar. Yes, alveolar. Because an acinus as a word stands for a grape. Yes, grape. And if you look at a grape that is still attached to a branch, you have it like that. Something like that. So this is like the tube or the ductal system, while this is the secretory unit. So if you look at that secretory unit, it appears rounded like a grape. That's made of acina, acinus. Alveolus, and alveolus is a sac. So even in the lungs, we have alveoli like that. So acinar glands are those glands where the secretory unit is like this, like a sac or like a grape. But tubular glands are those glands where the secretory unit is not like this. Instead, the secretory unit is usually like a tube. It is elongated. And then tubuloacinar, which you may also call tubuloalveolar, are those ones that have a tubular portion as well as an acinar portion. All of this is just secretory unit. We've not added the ductal system. So in the secretory unit alone, there is both an acinar portion and a tubular portion. And you'd remember that at some point in this lecture, I had drawn something like this that I described as a mixed gland. Yes, in this mixed gland, there's a tubular secretory unit as well as an acinar secretory unit. So here I may just come down to say that tubular glands, think of the mucous glands, for example. Mucous glands are tubular. Then acinar glands, usually we have the serous glands. So like the parotid gland that is serous is acinar. What about the pancreas? The exocrine part of the pancreas, it is also acinar. It is not tubular. And its secretion is serous. So serous glands are usually acinar. Mucous glands are usually tubular. So what about mixed glands that have both serous and mucous? those ones will be tubuloacina. So for tubuloacina or tubuloalveolar, our common example will be mixed glands like the submandibular and the sublingual gland. That's the much we'll take on glands. I would not want to bore you with more information. Like I said, there's the possibility of a fifth way of talking about glands. That's in terms of the ductal system, where we can have simple or branched, you understand? So all of those are unnecessary details for now, but hold on to these ones, read through these ones, and hopefully on the day of the exam you will not be lacking in anything. Remember to like, remember to subscribe, remember to share this video, remember to tell your friends about the YouTube channel Dr. Quinty, where they can find free videos on biology and chemistry. Enjoy your weekend.